Good day everyone, we are from Group 1. Here are the Group 1 members, ANISSA, ZHEN, ZHEN, NADIRA, DELINA and me, JING, and we are going to present about the mutation in SARS CoV-2. This is the infographic from our group which about the mutation in SARS CoV-2. Before go through the introduction, I will explain the definition. The definition of the mutation is an error in the cell genetic materials which alter the DNA sequence of an organism. The SARS-CoV-2 is the virus that causes a respiratory disease called coronavirus disease, which as we know as the COVID-19. Mutation SARS-CoV-2 is the permanent change in the virus genome during the replication. Now I will go through the introduction. In September 2020, the first new variant named Alpha was identified in the United Kingdom and is the first variant discovered by humans. The viruses with the new mutation are sometimes called variant. The variant can differ by one or multiple mutations. According to the research, the virus was evolving and this variant had a higher death risk compared to other variants. Based on the statistics of University of Oxford researchers, between 4,272 CCO patients, 62.8% were infected by Alpha and 15.5% died in the hospital. In addition, the infected person will have more self-symptoms such as fever, cough, shortness of breath, and so on. That's all from me, and I will pass to next group member to continue the presentation. Thank you.
Good day to Dr. Noor Shikin. My name is ZHEN. Now I will present about the mechanism and classification of mutated SARS-CoV-2. First, I will explain about the mechanism of the SARS-CoV-2 infected cells. So as you see the figure left hand side, the SARS-CoV-2 contains S protein. So this protein is the main structure that SARS-CoV-2 virus can enter the cell or infect the cell. So the RBD of S protein can bind to the ACE2 receptor, causing the viral mRNA infect host cells. This means that the SARS-CoV-2 infect the cells by binding the ACE2 receptor that found on the surface of the host cell, and then it will release the viral mRNA to infect the host cells. Once the mRNA enter the host cells, the translation will occur, causing synthesis more SARS-CoV-2 into the cells. And then the new synthesis SARS-CoV-2 virus will then release from the host cell and then infect another host cells again. So if the S protein is mutated, it will affect the ability of SARS-CoV-2 infect the cells. Generally, the mutated S protein will increase the ability or infection toward the host cells. Next, I will explain the classification of variants. So based on WHO, there are total two main classifications, VOI and VOC. VOI is variant of interest and VOC is variant of concern. So VOI is the genetic change of the virus are predicted or known to affect virus characteristics. This means that the mutation of the virus is already been predicted or already known, so we know the ability of the virus to infect the cell. The examples are Zeta, Eta, Theta, Lambda, and Kappa. For VOC, the definition is there is evidence of an increase in transmissibility and more severe disease. This means that this mutated virus will cause the transmissibility increase and also can result the severe disease. The examples are Alpha, Beta, Gamma, Delta, and Omicron. These are the famous type of variant under class VOC. So as mentioned before, Alpha, Beta, Gamma, Delta, and Omicron are the famous type of variant under VOC. So for Alpha, it is first found in late of 2020 in England, and there are total 17 mutations in viral genome. For Beta, it is first found in South Africa, and there are total 9 mutations in S protein and 3 mutations in RBD. It is same as Gamma, but Gamma is first found in Brazil on January 2021. For Delta, it is first found in India on December 2020, and it has 10 mutations in S protein. And lastly is for Omicron, and Omicron is the newest and dangerous variant now. It is first found in Botswana on November 2021, and there are more than 30 mutations in S14. That's all from me. Next, I will pass to Susan to explain the technique used to detect the mutation of SARS-CoV-2. Thank you.
My name is Susan. Today I will introduce about the technique to be used to detect the variant of COVID-19. We prefer to use multi-press quantitative RT-PCR than single-press RT-PCR because it is more effective and it can yield multiple results in a single test. And how it proceeds? First of all, we need to do a nasopharyngeal swab test. We insert cotton into our nostril to absorb the secretion about 15 minutes. The second step is collect specimen. Then we transfer the secretion to be stored under 2 to 8 degrees Celsius up to 72 hours. This helps to deactivate the virus. Therefore, we can proceed to RNA extraction. We just extract the purified RNA from the deactivated virus. After that, we transfer purified RNA to lab tubal DNAi or COVID-19 testing kit for reverse transcription and amplification by RT-PCR. The testing kit consists of reverse transcriptase, 3 target primer or probe mixture which are ZHEN, NGene, and RNAsP, as well as 2 type PCR master mix. After reverse transcription, the RNA will be converted to cDNA. Then we amplify it with 40 cycles. If the results show it across the threshold level within 40 cycles, this represents the positive COVID-19. In the RT-PCR, there is a screening system which are called VIR SNP probe-based melting curve assay will produce a fluorescent signals that represent the type of variants. This fluorescent signal will be detected and analyzed by light cycle software so that we can identify the variant present in the COVID-19.
Hi, I'm ANISSA and I'll explain the advantage of knowing the mutations of SARS-CoV-19. Of course, in general, there are no benefit for us if the virus itself undergoes various mutations. Hence, I'll explain this advantage in another point of view. Why should we be able to detect and classify these mutations in the first place? It is to have better understanding of transmissibility, disease severity, and efficiency of different vaccines towards different mutations. For example, the Delta variant is 40 to 60% more transmissible than the Alpha variant and roughly twice as transmissible as the original Wuhan strain. For disease severity, it is how sick you could get. For Omicron variant, it is most likely for someone to suffer all symptoms, such as fever, cough, headache, sore throat, and more. It is important to know the mutations for vaccines, since some vaccines cannot prevent us from getting reinfection if there are new mutations. Other than that, detecting these mutations earlier could help implementation of control measures to ensure vaccinated patients before these mutations are affected or not. There will be detection and surveillance of the virus, which will be followed by bioinformatic analysis that can identify lineage that increase in frequency or that it can affect previously immune individuals. And that is all from me.
Hi, I'm NADIRA and now I will be elaborating about the disadvantages of mutation in SARS-CoV-2, or you all might know as COVID-19. So number 1, variants of SARS-CoV-2 spreads at a faster rate than usual. According to an online article published last year, it was shown that one of the variants, specifically lineage B.1.1.7, was responsible for 28% of the COVID-19 cases in England itself. Not only that, this particular lineage spreads 56% faster compared to the other ones. Secondly, mutation in SARS-CoV-2 can cause false results on diagnostic tests. When a virus undergoes mutation, it creates variants which are genetically different from one another. This means that they have different RNA sequences. However, most molecular tests for COVID-19 can only detect a specific RNA sequence in the viral genome. Therefore, mutation causes this diagnostic test to be inaccurate. Moving on to the next point, mutation in SARS-CoV-2 can also make vaccines less efficient. The current vaccines work by releasing neutralizing antibodies when the presence of S proteins triggers it. Unfortunately, when the virus is mutated, there is a possibility that the neutralizing antibodies cannot recognize the variants of the S protein. And last but not least, certain SARS-CoV-2 mutations can cause an increase in hospitalization rates and also death rates. Tests were done in the UK community test centres, and it was stated that individuals who were infected with the alpha variant were between 32 to 104% more likely to die compared to previous variants. According to another study done by independent researchers, they concluded that a mutation in the ORF3 protein could increase the mortality rate of COVID-19 infection.
Next, let's move to the effect of SARS-CoV-2. Based on statistics, the SARS-CoV-2 pandemic has infected over 2 million individuals across the world, resulting in 4,752,899 fatalities and significant disruption to regular activities and national economies. It shows where national economies dropped down to its lower points during the pandemic gradually. And then let's move to the second effect, which is policy of government. The government has established the movement control order to diminish the number of new cases per day. Example for the movement control order that has been established is wearing masks, social distancing, normalize using hand sanitizer, and no big event. Next, for social, limited movement to the public places, early closing hours for business sectors, working and classes from home. As for the business sectors, as I mentioned just now, it affects for the national economies. And socially, it affects regular activities.
So, let me to conclude our presentation. In a nutshell, the mutation in SARS-CoV-2 that caused the COVID-19 has undergone rapidly in our world. Although some variants have mild symptoms, there are also variants with more infections and also resist the vaccine. Hence, we must get vaccinated and have a good personal hygiene to reduce the spread of COVID-19 and get back to our normal life soon. That's all from our group. Thank you.